Welcome everyone to this virtual question and answer forum on COVID-19 brought to you by the New South Wales Smart Sensing Network. I'm Professor Justin Gooding from the University of New South Wales and the co-director of the New South Wales Smart Sensing Network. The Smart Sensing Network connects industry, government and end users who need new types of sensors to university researchers that have the capability to develop those new sensors. This is clearly relevant to the pandemic we're all facing. So with my co-director, Professor Ben Eggleton from the University of Sydney, we decided to put on this forum because in these constantly changing times, we, like so many of you, feel very uncertain. And we, like so many of you, really want to do the right things by ourselves and the right things by the community to limit the spread of this virus. And with the constantly evolving information that we receive, we, like so many of you, feel quite concerned, quite confused, and we're unsure what is the right thing to do. And so we assembled a panel of experts to answer all your questions, whether they be very simple, practical things like how often do I need to wash my hands, or more scientific things such as how does the virus enter a cell. And with that, I'll hand over to your moderator, Robyn Williams. Robyn Williams is the long-running host of the ABC Science Show here in Australia and an extraordinaire at making science accessible to all. Over to you, Robyn.
Thank you very much indeed. Yes, I've been doing the Science Show for 45 years now. A long time. And as you heard, we're inviting questions from you, the audience, complicated questions, simple questions, and we encourage you to write in, and those questions will be looked at by various people we have lined up, and they'll be selected and we'll give answers a little later. And this forum will in fact be given live, broadcast live, streamed on Sky News COVID-19 on this weekend. Just to give you an example of some of the questions, the style, I asked a strange question the other day because I often have sandwiches with my newspapers. So I said, are the newspapers in any way germy? Are the viruses living there? So that every time I turn the page and lift my food, is there a problem? Now, it just so happened that yesterday, Peter Doherty, our Nobel Prize winning scientist, answered that question in a way by saying if you've got a pizza box, the virus can live on the cardboard outside for a day. So if you order pizza delivered to your door, then you open the box and lift out the food without touching it, and then throw the box away, and then maybe you eat. Another question about chloroquine, which seems to be rather famous. Some people are suggesting that it's possibly a medical answer in the short term. And finally, I had a question before I left home this morning about whether you can wash those PPEs, those personal protective equipment clothings. And the answer is probably not. Anyway, we have a panel waiting for you and your questions. And they include Professor Bill Rawlinson, a virologist from New South Wales Health. Professor Justin Gooding, you've just met. His special field is rapid sensing of various germs. Professor Sacha Stelzer-Braid, respiratory disease in the vulnerable from the Prince of Wales Hospital. Professor Mary Louise McClure, epidemiologist, who examined the response to the outbreak and the infection prevention during SARS 2003. Remember that one, we should have learned more, that outbreak. And she was a member of the WHO advisory panel for COVID-19. Professor Paula Store from University of New South Wales, who knows all about soap. And we also have some virtual experts online. Associate Professor Nathan Bartlett, University of Newcastle. Professor Robyn Williams from the University of New South Wales. Professor Paula Store from the University of Newcastle. Professor Guy Marks, President of the Union. Associate Professor Gary Myers from the University of Technology in Sydney. Professor Subhas Mante from Macquarie University, and indeed Professor Antonio Tricoli from the ANU, the Australian National University. Now it's my pleasure to introduce Professor Bill Rawlinson, Senior Medical Virologist and Director of Virology at New South Wales Health, to give the keynote presentation. Bill.
Thank you very much, Robyn, and welcome from me too. I'm going to do a brief introduction. It falls to me to really introduce the subject which I guess many people have heard about. You'd have to really not be awake to have missed what we now call COVID-19, which is the disease, which is due to a virus called SARS-CoV-2. It's called this because of its close relationship to SARS virus, which was first described in 2003, causing the outbreak of severe acute respiratory syndrome, hence the acronym. So I thought maybe the best way was to have an introduction that listed sort of some of the things that might be questions, and then answer them, and perhaps address some of the things that you might like to ask later. The first thing is that people often sort of think about this as a presentation of a patient with COVID-19 or a patient with the virus, which we immediately see and then we can do something about it. But of course, the reality is it's called severe acute respiratory syndrome because it is a syndrome. It's not a specific disease in the sense that you can recognise it easily. And this is a slide that Sacha Stelzer-Braid prepared. And this is really some of the viruses that cause respiratory infections. And people are very familiar with influenza A and influenza B. They occur as epidemics every year. They can cause a mild illness or they can cause a severe illness. And of course, we have vaccines and antivirals for influenza. But it's also important to realise there's a whole range of other viruses listed here, which typically give an upper respiratory infection. So sniffly nose, sore throat. Sometimes they cause a fever, such as adenoviruses. Sometimes they cause conjunctivitis, such as adenovirus. But they're often indistinguishable from each other. And then there are the coronaviruses shown here in red. And they cause typically every year episodes of about 1 in 10, 1 in 8 of all the upper respiratory infections you get. So all the colds, about 1 in 8 of them each year are due to coronaviruses. This is not what we're going to focus on today. These are the other ones. These don't cause pneumonia. They don't cause people to die. This is the sort of problem down the bottom in these chest x-rays. The one that I'm showing here on the left, that's a normal chest x-ray. The one on the right is one that's significantly abnormal, and this is a viral pneumonitis that's well advanced, and this is in fact in a patient in ICU. And this is a sort of disease that you can get potentially from SARS coronavirus 2. This is the worst outcome and sometimes results in mortality. You don't tend to get this with these other viruses, except perhaps for influenza and sometimes with para-influenza. And this just shows at the bottom the months from January through to December, and the number of cases from things like influenza. They tend to concentrate very much in winter. Some of the other viruses I showed you are spread more throughout the year, like adenoviruses. And here are the coronaviruses. Again, typically a winter disease, but in Australia spread somewhere between around about March, April through to about October. And these are the other non-SARS coronaviruses in Australia. So the virus that we're talking about, SARS-CoV-2, arose first in Wuhan in China in a wet market, and then it spread nationally, of course, as you know, has spread internationally. There's clearly been some discussion about this, but the early epidemiology, and this is a paper from a Chinese group who published just this year, looking at cases before January the 1st and then up to the end of January. And basically at the start of January, more than half of these cases were associated with a specific market in Wuhan in Hebei province. And as the outbreak progressed, increased numbers of those were not from Wuhan, but from other parts of China. Importantly, I think from all of our points of view, in terms of going forward, early on, these were people working the markets, these were people on the streets. They were not healthcare workers. But as it progressed and as healthcare workers cared for some of these patients, the number of infections of healthcare workers went up. And indeed, that's something that concerns us all, not only for our sakes, but also because what that has resulted in some cases has been a lack of healthcare workers available to look after patients. the virus SARS-CoV-2 is very close genetically, so its genome, its genetic structure, is very similar to the other coronaviruses. It's also more similar to bat coronaviruses, and in fact, it's very closely related to the SARS coronavirus that caused infections during 2003. And this just demonstrates, I guess, schematically, that a whole lot of coronaviruses occur. They occur in bats, rats. They can be transmitted to a number of other animals, alpacas, cows, sheep, pigs, camels. And these viruses clearly are able to jump between these species, and of course, in some cases, can jump into humans. And so when we look at these viruses and we think about them, we call them zoonoses, that is animal-derived viruses. And that may be through an intermediate host. And here's a civet cat. That was the intermediate host, we believe, for SARS, although the viruses that we see now are really very closely related to bats. There's been some discussion about snakes as being an intermediate. That's actually wrong. These viruses that are infecting humans are most closely related to bat coronaviruses and may well be a direct jump from a bat to a human. When we look at SARS coronavirus 2, of course, things are getting updated daily and almost hourly. As of yesterday, there were almost 700,000 cases globally with 35,000 deaths. There were just over 4,000 cases in Australia with 16 deaths, I believe, as of this morning, that's 18 deaths. And of course, it's now spread globally. And to put it in perspective, even though genetically SARS coronavirus 2 looks like SARS, the SARS virus we saw in 2003 infected a total of about 8,500 people. And in fact, we saw no cases in Australia. There was one individual who was a backpacker who travelled back to Germany and was diagnosed there. So what we're looking at now, even though it's similar at a virus level, even though it looks the same genetically, it's a very differently acting virus. And there's many reasons for that. And I'm sure we'll discuss that as we go through. So the progress, this is first a map locally, and here's a global map. Looking at the global map on the left, you can see that Australia has this kind of reasonably high rate of cases. These are the rates of COVID-19 disease per 100,000 of population. And the rates seen in Australia are similar to those seen in parts of Europe, as well as in the US, but much higher than rates seen in places like Africa. And of course, there are still questions about the rates in countries like Africa and India because of the low rates of testing and some of the lower rates of access. This is where I work, which is the eastern part of Sydney, and these black dots represent, as of 2 days ago, the number of cases that are being diagnosed. Where I work, which is at Randwick for New South Wales Health Pathology, we have the largest concentration of cases in the state, and in fact, Waverley and Wallara, which are suburbs close to us, have the largest rates of diagnosis of SARS coronavirus 2 and COVID-19 currently in the state. And in fact, New South Wales has the largest number of cases in the country. The way we diagnose SARS coronavirus 2 is very similar to the way we diagnose all viral disease now, which is using molecular tests. And some of you may have heard of the polymerase chain reaction, or PCR, but basically it's a type of molecular test which detects the genome of the virus. It's an assay that's been developed over the last couple of months for this specific virus, but PCR tests, these molecular tests, have been available for over 25 years. We also increasingly look for antibody using serology, and that can tell you if somebody has had an infection in the past. So it was suggested I might just go through the process of diagnosing SARS-CoV-2, which is similar to the process of diagnosing any other respiratory illness, except that the large numbers and the significant concern about spread to individuals from close contact means that we're very concerned about protection of our healthcare workers, protection of our collectors and other people involved in the chain. So the red strokes indicate how not to do it. And this is kind of how we do it. The only thing that's wrong here is the individual taking the throat swab would take a throat and a nose swab and should have a gown on, but correctly has a mask. We do use visors rather than glasses these days, and we do use gloves. clearly not having a mask and clearly hurting the patient is not what we aim to do. Importantly, there are also specific clinics where such testing is done, and the healthcare workers there are very familiar with how to protect themselves from infection by the patient. That is what we call PPE, personal protective equipment. And that's obviously very important for us as healthcare workers, but it's also very important that we don't get infected and then go ahead and infect patients in settings where we may not be using correct PPE. The other point is that the specimens are transported in a fairly normal manner. We regard this as a fairly low-risk organism when it's handled correctly. Clearly, we don't put it onto motorbikes because of the risk of spillage and damage. And clearly, when we handle it, we don't regard this kind of protection as necessary because the specimen is carefully packed into appropriate materials so that it can't spread and, in fact, it can't be damaged. Rather surprisingly, I guess, this step from collection through to handling in our laboratory is probably the longest step. The actual testing downstream currently takes about 3.5 hours, but it's really handling the specimen and receiving the specimen that can take up to 8 or even 12 hours. And so often it's those logistics, just the handling of it, that takes a lot of time. So people often say, well, you've got now tests that you can do sometimes in 3 or 4 hours. You've got tests, in fact, coming online, which you can probably do in about half to 1 hour. Why can't I get my result immediately? It's really this chain. It's getting from the individual who's being swabbed through to our laboratory, which is shown here, that takes the time. There's a lot of tests. This is one we use. This is another one we use. They're automated. So what that means is you can load them and walk away. And of course, the results ultimately come out electronically. And so the process of diagnosis now is really quite smooth. And our job is to reduce that time to get it done quickly. Our laboratories, the Serology and Virology division, we commenced testing very soon after the genome of the virus was made available by the Chinese online, who had taken the virus and within about a week had determined the complete sequence. As I mentioned, we utilised this molecular test, which many of you know, called PCR, initially using assays that we made up in the laboratory, and these are the the staff, the scientists who are doing the work at the moment. And increasingly now there are commercial assays, so companies have come on board. In fact, there's now 40 or 50 that I know about. We use a couple of those, the reason being that we build in redundancy because we have reduced mean time between failures, because as you increase the number of tests you put through on a platform, the number of breakdowns, the number of problems arise. And really our job is to take care of these kind of issues like shortages of reagents, breakdown of equipment, things like illness occurring in the staff, and also having plans so that if one of us gets sick, if one of us, for example, develops infection with the SARS-CoV-2, that there's still others to work. And so, in fact, we're all holding our breath during this photo and then ran back down the corridor because these people are broken up into 2 teams, and those teams, for example, have separate lunch breaks. They don't work directly together during the day. And so that's the general introduction I'd like to make. Fred Sanger, who many of you know, now sadly died, who received the Nobel Prize twice, developed DNA and protein sequencing. This is one of his comments from 40 years ago now, which I think is quite prescient through art and science in their broader sense. Senses it's possible to make a permanent contribution towards the improvement and enrichment of human life. And it's these pursuits we students are engaged in. And of course, at this stage, he was a world famous Nobel Prize winner. And I think to regard ourselves as learning and as students is part of this, and this is what the outbreaks taught us. So thanks very much.
Thank you very much, Bill. Yes, Fred Sanger, one of the most modest people I've ever come across. 2 Nobel Prizes, but they didn't go to his head in that respect. Now I'm going to ask most of the people we have here, the panel and the experts online, just to give a couple of minutes summary of their own particular expertise, starting with our host, Justin Gooding, and something you hinted at, Bill, rapid sensing.
Yes.
Justin.
Hello, everybody. I'm Justin Gooding, as you've already met. My expertise is rapid sensing and ultra-sensitive sensing. And I actually have a question for Bill for later on, which is, what would be the dream diagnostic from your perspective? But I'll pass on now.
Next, Professor Sacha Stelzer-Braid, and your particular expertise.
Hi everyone, my name's Sascha and I'm a virologist working in the Virology Research Lab at the Prince of Wales Hospital and also at the University of New South Wales, and I'm interested in respiratory viral infections, particularly in people with chronic respiratory diseases.
Next, Professor Louise McClure, please. Epidemiology. Louise, are you there?
Very well. Hello. There we go. Sorry, I was having trouble with my unmute. I'm just an epidemiologist. I'm an epidemiologist. My area of expertise is infection prevention and control. I work at the University of New South Wales, but I also have worked for the World Health Organization, and I'm on the COVID-19 expert panel in trying to identify safety measures for healthcare workers and the public. Over.
Now, Paula Store, soap, I understand.
Yes, hello. Yeah, my name is Pat Thorson. I'm a professor here in chemistry at the UNSW and also part of the Australian Centre for NanoMedicine and the Centre of Excellence in Bio-Nanoscience. And I work on a self-assembly of molecules into functional materials, including nanoparticles, and use ampliphiles, which is really like soap molecules, to make nanoparticles, which essentially viruses are. Viruses are nanoparticle, and that sort of dragged me into this discussion over the last few weeks.
Now a few words from our virtual panel, starting with Professor Nathan Butler. Nathan Bartlett, are you there?
Mute me. There we go. Hi, I'm Nathan. I run a lab at the Hunter Medical Research Institute, part of the University of Newcastle, and we've been researching the really early events that occur when a virus infects your airways. We believe understanding those really early events are going to be key to the development of really effective treatments for respiratory viruses such as SARS-CoV-2.
Next, Dr. Ron Bouveray.
Yeah, hello everyone, I'm Ron. I help researchers at the University of New South Wales to commercialize a new technology that addresses exactly the problems that Bill spoke about, the long diagnosis process. And ideally, we would like to develop a technology that delivers a fast, quick, specific diagnostic on the spot. Imagine someone that could deliver diagnosis in a corridor.
Next, Professor Paula Store, and you're involved with those test kits and masks and so on. Paul.
That's right, Robyn. Good morning, everyone. I'm Paula Store. I'm a professor of physics here at the University of Newcastle, and we're involved in developing large-scale manufacture and printing of test kits, and we're also now involved in developing PPE at large scale for our hospitals and GPs.
Next, Professor Guy Marks, implications of COVID-19 epidemic in low middle income countries.
Hi, I'm Guy Marks. I'm a respiratory physician and an epidemiologist. I work at Liverpool Hospital in Sydney, but I'm also the president of the International Union Against Tuberculosis and Lung Disease. I do work on tuberculosis and other chronic lung disease, in around the world, I guess. And I'm particularly interested in the burden that this disease is going to place on countries with already very stretched healthcare systems, and how they're going to cope or are coping with this pandemic.
Professor Subhas.
Hello everyone, I'm Subhas. I work at McClure University. I'm a professor here on mechatronics engineering. In my research, I develop different selective chemical and biosensor, especially for bacterial infection and so on, which is basically fast detecting device.
Professor Antonio Tricoli, biomedical sensing. How does that work?
Good morning, everyone. I'm a professor of engineering at the Australian National University in Canberra, For the past 15 years, I've been working on developing miniaturized biomedical sensing device for point of care and wearable sensing technology. And I've done this both at PTH Zurich in Switzerland and the Australian National University. And one of the devices that we have developed is about actually using what comes out of your breath for diagnosis of diseases like diabetes. And we work also on minimal invasive technology. So basically trying to diagnose diseases by a small amount of blood that you can take from a fingertip.
Thank you very much, panel and experts. Let's go to our first question. It's from Maria. I'm not sure where she's from. In future, could you indicate where you are, if you're somewhere in Iceland or somewhere in New South Wales? Just one little indication of your geography. But Maria says, if I go outside and happen to have the virus living on my clothes, can I place the clothes in direct sunlight to sterilize them? Now, I think 3 of our panel have an answer. Let's go first to who volunteers.
Hi, I'll volunteer first.
Thank you.
First of all, let me allay your anxiety about the possibility of having the virus on your clothes. There's just been a paper recently published, it's a small one, of 13 healthcare workers who looked after patients with COVID-19. They tested all their personal protective equipment, including their shoes. And you'd expect them to be covered with the virus, but they found it on their shoes, but not their mask or their gown or their gloves necessarily. So good news is it's unlikely that you'll have virus on your clothes, although Bill Rawlinson, myself, and a PhD student we were supervising did check clothes of kindergarten teachers, and they had rhinovirus and other viruses on them, but whether or not being. So I'd suggest that you're quite safe putting your clothes in the sun, or if you could, because this is what we suggest to the healthcare workers with their linen, is to wash it and add some bleach. If you can't add bleach, of course, a soap wash should be just fine. But I'll leave that to Paul because he's pretty good at soap. So over to you.
Paul.
Yes, thank you, Mary Louise. Thank you. And this just confirms what I thought with clothing probably not be a very good carrier, because I don't think these nanoparticles and viruses stick very well to the fiber, or if they do, they're so buried inside you can't pick them up again. And I'm absolutely with you that if you are unsure, just wash the clothes. Sticking them out in the sun might help. There are indications that similar viruses don't tolerate sunlight for long, but I'm not sure that's a very effective strategy in this case, because you would still need it light for some hours possibly, and you may not get to the other side of your clothes. So I would wash them rather than trying to use sunlight here.
Paul, by the way, the question I asked at the beginning about paper, we get not just clothes, but paper is ubiquitous, and we read the newspapers and magazines or whatever. If you're not a screen person so much, but even screens may be covered. But the paper, is there much evidence? I know I mentioned Peter Doherty talking about how on your pizza container you can have the virus living for a day.
Maybe someone else can chop in here. I've only found one paper that did some virology on paper, and it was not much better or worse than other common surfaces like steel. So I think we need to treat paper as a potential fomite. But my advice would be, wash your hands, take the pizza out, throw the box away, and wash your hands again.
Right, thank you.
And now you are probably safe.
Splendid. Question 2 from Aran. There were some claims by some Indian researchers and doctors successfully treating COVID-19 patients with HIV medicines. Does anyone on the panel know about it and put light on this, if this could be a potential line of treatment?
So, Robyn, perhaps I'll take that to start.
Thank you.
There's trials of antiviral protease inhibitors called lopinavir, ritonavir. It's a combination called Cletra. And those trials were actually done way back in SARS time. So there were initially some studies where SARS-1 was looked at. And so that's led to a number of ongoing studies, not only in India, but also in China and in the US, looking at lopinavir-ritonavir combination, as well as other antivirals, as well as some other things such as immunoglobulin from recovered patients. Now, there's a number of reasons those drugs might work, and there's a number of reasons they might not. But in short, the initial review suggested that they didn't show any benefit, although there's been some more recent studies just in the last 2 to 3 weeks where there has some patients who received lopinavir-ritonavir in combination did better.
Now we're after question 3. Comes from Ken from Roselle in New South Wales. If soap is so effective in killing the virus, why can't they make a soap safe for drinking? Ali?
Well, the short answer, we just can't. So soap is always dangerous to drink. It will irritate your your mouth and your stomach and whatnot. So it will be extremely dangerous to drink, digest, or spray soap into your body. But I'm going to pull this over to Nathan also to explain why it wouldn't really work anyway.
Nathan?
Yeah, look, I mean, for a start, drinking something delivers it to the gastrointestinal tract, and if you're trying to target a virus infecting your respiratory tract, then that's not likely to be particularly effective. And the other major issue is the fact that the virus is an intracellular pathogen, so once it gets into your cells, you need to kill the cells that are harboring the virus in order to eradicate the virus. And soap is not particularly effective at doing that in any sort of controlled way. And as we just heard, it would severely irritate or be toxic to the more delicate lining of the airways. So yeah, it just wouldn't work. It would cause you more damage than any possible good.
So no drinking soap, I'm afraid.
No.
It's an active ingredient, who knows, but no. Next question. Why do we treat SARS-CoV-2 so differently from the other coronaviruses? Why are we so careful and cannot use the knowledge we gained from the other coronaviruses? That's from Romaric Bouveret.
Perhaps I'll start.
Thanks, Bill.
So even though genetically SARS-CoV-2 is like those other ones I showed, NL63 and the others, it is very different to them clinically. So it's also different to SARS-CoV-1. It seems to be somewhere in between in terms of mortality. The other coronaviruses don't kill people, and SARS-CoV-1 seemed to have a mortality of about 9%, so about 1 in 11. So it spreads in similar ways to the other coronaviruses, but it seems to cause a more severe illness in some people, and that's particularly the elderly. And it can cause pneumonia in a larger number who don't die, but of course need intensive care in some cases. So it does seem that it's genetically similar, but probably because it's come from directly from animals, it's a zoonosis. It's meant that it causes more inflammation, more fluid, if you like, in the lungs, which causes pneumonia and hence causes more severe disease. What we have learned, of course, from the original SARS is that some of those drugs that I mentioned and some of those vaccines and some of those procedures are carrying over into treating and diagnosing this one.
Bill, is it possible that people who are, shall we say, over 70 can get a certain resistance by being fit? In other words, doing exercise, having an immune system that's bolstered?
It's a very good question, and clearly if you're unfit, so if you're over 70 and have heart disease or lung disease, you're more at risk from severe disease, so with SARS-CoV-2. So the other side of the question is if you have those illnesses, then you are more likely to get pneumonia, and in fact you're more likely to die. So in Australia, if you're a normal fit over 70-year-old who has regular exercise and a diet, then that's as good as you can do in terms of your immunity. There's a lot of discussion about other therapeutics and other things you might have. So far, there's been no evidence for those.
Thank you. This question will go number 4 to Mary Louise McClure's, and then anyone else. It's question 4 from Ben from Camperdown and the NSSN co-director. The government seems to suggest at some point that kids don't transmit the virus. Is there an update on this point? Seems unlikely.
Hello. Yes. Hi, Ben. Thank you for that question, because we are getting a lot of mixed messages. Still, the children are rare numbers in Australia and around the world. In China, they represented less than a couple of % of all cases. And in Australia, it's no different. Bill Rawlinson may want to talk to you about the potential role of ACE2 receptor sites, but we're a little clueless at the moment about why this is, but we don't believe that the children drive the epidemic. Thank you.
Anyone else want to add about kids?
Maybe just to make a comment about kids, that they don't seem, fortunately, to be getting severe disease. It often gets asked, why is this not like influenza? One of the big differences is that in influenza, the very young, as well as the very old, can have severe disease and in fact die from influenza. So far in SARS-CoV-2, we've seen a very small number of people under the age of 18 with severe infection. There have been, just in the last 24 hours, a couple of deaths, but in terms of the hundreds of thousands of infected, there have been very few severely affected children, and certainly under the age of 9, nobody so far who's been severely affected. So it does seem to act differently in kids to some of the other viruses.
Well, question 5 from Fong from the University of New South Wales. If the SARS-CoV-2 mutates quickly, would a vaccine still work? Well, Sascha, what do you think of that? You know, we get a vaccine in a year's time and doing, will it still work, the mutation?
Question. And SARS-CoV-2 is an RNA virus, and what that means is that what we know about RNA viruses is that they do mutate fairly rapidly compared to viruses, and a good example of that is influenza. So we know every year we have epidemics of influenza because the influenza has the ability to mutate over the season. And so know at the moment about SARS-CoV-2, but we're doing a lot of research to try and work out how quickly the virus is changing. And I would think that if we have a vaccine available, then that vaccine would either...
We've lost you. It's a question about whether you can stay one...
I'll take like 50 to 60% protection. That vaccine is quite good and offers sort of... Sorry, what was that?
Yes, we lost you for a bit.
Oh, sorry. Okay.
Well, what you seem to be saying is something like what we do with the flu vaccine. We try to predict what the next season of flu molecule is going to be like and stay one step ahead, so we're waiting for it. So even though the COVID may mutate, nonetheless, you're halfway there to getting something that could answer for strain 2. Is that more or less what you're saying?
Yeah, that's exactly right, Robyn. And it might be the case that, you know, we need to monitor how the virus is changing and sort of update our vaccines. But we really have to wait and see with that.
A technical question while you're there. As it's RNA, in other words, the version of coding from DNA transmits, but does the virus reproduce in the same way by taking over the genetics of our own cells, which then get sort of co-opted to reproduce the virus for it?
Yeah, that's right. So, and that's what all viruses do. So they can't reproduce by themselves outside of our bodies. So they're only replicating and they're only really alive when they're infecting our own cells.
Super. Let's go to question 7 then. Sorry, does someone want to come in? Okay, moving on. Question 7 from Simon. I'm watching from Melbourne, he says, from a question for Justin, that is. He would like to ask Justin, is there any electrochemical biosensor being used for COVID-19 detection in Australia? Any perspectives, Justin?
It's actually a really good question, Simon. I'm going to make it a little bit broader than that, but actually Sascha and Bill and myself just put in a grant application with a Melbourne-based company that's developing electrochemical sensors for doing exactly that. And what the sensor's designed to do is actually detect the antibodies that your body raises to fight the virus. And this would be then a portable device that you could use or could be used in a doctor's surgery. The real advantage of that is that when Bill showed you that chain of steps that they have to go through to bring a sample to the lab, you could avoid that side of things and give a quick answer. But even then, you would need another technology, a technology like PCR, to verify those results. And so actually Bill and Sascha and myself also worked together on an optical device that would allow them to very rapidly detect the viral RNA very rapidly without requiring the amplification. But I think that we have other people here, like Paul Store may have something to add here, or Antonio as well, because they're also working in this space.
Yes, please. Paul.
Thanks, Justin. Yes, we've just been funded to work together with Nathan, actually, on developing a rapid test for COVID-19. It's going to be based around RNA switches, and we will be focused on developing the sort of instruments that will do the test. So our expertise is in rapid prototyping of medical devices. I might throw to Nathan to talk a little bit about the mechanism of detection.
Nathan.
Yeah, so it really is, it's based around detecting the virus's genetic material, the RNA that you've alluded to. And when we can use, we've got infection models with related coronaviruses. So you heard about the other coronaviruses that were discussed by Justin earlier this morning. So we can use those as a good prototype to show that just by have a small sample of infected material, we can use these approaches to directly measure the presence of the viral genetic material, and using a very complicated process that I don't fully understand, quickly and efficiently identify the presence of that viral genetic material, and that's as a diagnostic.
Talking about quickly, do you mean overnight, 24 hours?
I believe it's within an hour. I think
an hour.
Yeah, within an hour is the aim, yes.
That's impressive, yeah. Next question from Inca in Brisbane, who says, I see people wearing more and more homemade fashionable fabric masks. Are these at all helpful protecting yourself from the virus? Let's say, ask Paul Store. Fashionable masks.
What we have to do is to create an effective barrier to transmission of the virus. We have been rapidly developing prototype face shields since the beginning of this week, actually. And we now have those being manufactured here at the university. And if I just might share a screen just to show you what we're doing. We are building these devices in our clean rooms. We are then, let's just share that screen. If you've got that. And then this is the sort of device that we've produced that's going out to emergency departments, to hospitals, and to GPs at the moment for some feedback, and we're now working out how to scale that up.
Splendid.
Robyn, can I take this question? as well?
Please.
Okay, so WHO is very interested in masks and the type of masks that can protect healthcare workers. And then, of course, what we find will also advise the public because the WHO and every other country in the world is worried about the supply chain. So we're very pleased to see that mask production is occurring in Australia. There's been a systematic review of different mask types, and cloth masks are found to not protect against influenza A and B. And those fashionable masks are unlikely, but I can be corrected, but they're unlikely to protect you because they're not made of the very complex fabric and the charge that they need within the fabrics to ensure that the filtration is greater than 98% of the most penetrating particle. And strangely enough, that penetrating test particle is actually a salt particle that they test in the lab. And if that can't get through, then it's unlikely that particles such as COVID-19 can get through. And I doubt that your fashionable mask has been tested for that. Over. Thank you.
May I just ask you a question, Mary Louise, to follow that up? Something that came into me personally from a person I know about the washing of the PPE, that's the personal equipments. It was suggested that the masks, if washed, would simply fall apart and be non-functional, but maybe some of the other equipment, which is running short, could be washed. Have you an opinion on that?
Yes. Look, as a part of the WHO panel, we have looked at this because some countries are incredibly anxious about their supply chain. And it appears that washing a mask, such as a surgical or otherwise called a medical mask, it will degrade the strands inside the masks. So don't do that. Certainly, if you feel comfortable wearing a cloth mask, but you wouldn't want to wear a cloth mask in a hospital, you'd need to clean it. And that may protect you from a lot of things, but it probably won't protect you from COVID-19. And certainly we don't recommend washing surgical or medical masks for either the healthcare workers or the public.
We have another question about people who've recovered, supposedly. Are they fine to go out in the general populace eventually?
I'll make a comment. Sorry.
I'm thinking about Prince Charles.
No, okay, Bill, you go first.
So, sorry to interrupt. So we've recently drafted documents around this in the Commonwealth level and at the diagnostic laboratory because the test we do detects RNA, not live virus. And so the recommendation is that once somebody has recovered and it's more than 14 days and they have 2 sequential days when they have negative tests, then we would regard them as not infectious and that person is in the community is not infectious. Sometimes those tests can become positive again, and that's been in the literature and in the sort of chat. That's most likely because it's non-infectious RNA. I mentioned the genome is present. It's an RNA genome and we can still detect it, but we think that virus doesn't grow. So if we try and infect with that virus, it actually doesn't work. It's just the RNA, not the whole viral particle.
We have a question from Kathy. If rapid testing targets antibodies, how will the introduction of rapid testing improve detection rates, and will it help with monitoring community transition? Antonio, what do you think?
Well, I think this is actually an excellent question. It's mostly about what we are trying to understand and target. The knowledge so far is that this rapid testing of antibody is effective at least 14 days after the virus has been active. So it will not give you an immediate information of whether you are actually contagious for others or not. Now, what could be useful for is like once you had the virus to know if you're safe to go out and if you're still can infect other people. The problem with all these tests then becomes accuracy, right? What if you have like a false negative or false positive? So I think at this stage, there is great potential to bring people back from home self-quarantine to work and to the city. This is something necessary. But I guess there is also still a little bit of knowledge required to know how precise and accurate these tests are.
Thank you. Anybody want to add? Let's get a question 10 from Sarah in regards to cleaning. What measures should we be taking at home for those working, for instance, in supermarkets or teaching? I take it that HCW know what to do versus those just going out for essential items. In other words, particular cleaning approaches to people for people in particular jobs. Pali?
Sorry, I thought Mary Louise was going to do this first. There have been quite a lot of studies in the past on similar viruses in terms of what works in terms of cleaning, and this one, being a really delicate virus, seems to be, you can probably kill it with the most cleaning products. The debate that's going on, and there's a little bit of debate, should we focus on bleach-based products when you have them, or should you actually clean with detergent? There's some evidence that, and I've seen papers suggesting cleaning detergents, the normal ones you use on your floor and whatnot, will kill these sort of envelope viruses. And the other argument for using those is that you clean the dirt away as well. And dirt might be a place where the virus hides. And if you just spray with bleach, you probably kill all the viruses on the surface, but if it's a dirty surface, it might not be sufficient. But I think probably a mixed approach is best. And I would like Mary Louise to comment on this because it's a hot topic.
Mary Louise.
Hi. So from the WHO perspective, they have, you can get it on the web. They have a list of products, and Pali is correct. It starts off with soap and water, but before then, the step is to remove visible dirt. And so then you do a soap and water wash. But if you want a bit more of a clean, you can use a 0.1% bleach, and that's quite a heavy bleach solution. That's normally for hospitals or hospital in the home. If you're just in the home, just use a very weak bleach solution that won't ruin your metal, and that should be more than enough. Hand hygiene when you come home from shopping, and hand hygiene before you take your shopping out and put it into the fridge, and then hand hygiene later. And then you should be fine. You'll be in your own bubble of your own germs at home.
Thank you, Mary Louise. Question from Mike in Adelaide. We've heard about the efficacy of molecular tests, but wouldn't it be easier just to do mass temperature testing of the population? Subhas, what do you think? Subhas? Are you muted? Well, we're coming to Subhas. Anybody else want to comment?
Can you hear me?
Oh, there you are, Subhas. Thank you.
Yes, the temperature testing is the common method used when people need to test very quickly. So it's used at the airport and other places. But temperature only will not give the assurance of that person's suffering from COVID-19. So if we want to do rigorous testing, especially we need to see whether the breathing rate, sore throat, runny nose, so many other symptoms come to picture. So temperature is the only way to go, just to have very quick test, and that's what people do at the airport and other places.
Anyone else want to comment?
Just to make the comment that not all patients with COVID-19 have a fever, and some of them don't develop any fever and are still infectious. We think that in those who do have symptoms, fever is present in about 90%, but it's very likely there's a large proportion of the population who we're not testing, who are not developing a fever or developing a very low grade temperature.
Thank you, Bill. Next.
Can I add to that? Just
please,
very
yes.
briefly. So on the borders into China during SARS in 2003, they had the testing. And one of the problems is that people can move around and take a paracetamol to lower their temperature so they can move from one country to another. So you can't necessarily accept a temperature screen as screening out or ruling in people with disease. And as Bill has mentioned, not everyone elicits a temperature, and particularly during the Beijing outbreak, we looked at the definitions, and when they expected people to have a temperature, we missed many SARS cases because people who have multiple comorbidities often don't elicit a temperature. But temperature testing looks good. It sends a really good social message that you're taking care of your citizens and that you expect them to do the right thing by the other citizens as well, by not moving around society with potentially an infection.
Thank you, Mary Louise. Now a question from Sarah in Hobart. She says, we have seen these terrible images of people collapsing in the streets in Indonesia. Are the challenges different in managing this virus in low to middle income countries versus countries such as our own, better off? Guy, what do you think?
Well, clearly there are additional challenges in low and middle income countries which make it even more difficult than it is in our countries, and the lack of sophisticated health infrastructure to deal with patients once they develop the virus, the lack of ventilators, intensive care, and so on is a huge problem. That's on the one hand. On the other hand, what is really key to controlling this virus is in fact public health interventions that have been available to us for centuries and don't require much technology. It's really quarantining, isolation, social distancing, and so on, which in fact don't require a lot of technology. So what we need to do, I think, in countries which don't have the technology and the capacity, is to try and build capacity for those sort of simple, low-tech interventions, which will help to control the virus. But the other side of that coin is that whereas social distancing and everything is having a major economic impact in our countries, we have more resilience to that economic impact than do poor countries that are already very poor. So you can imagine that if people can't get out to do their normal activities, that has an even greater impact on people who are living in poor countries. So yes, this is going to be an absolute disaster. The bottom line is this is going to be a disaster, but there are strategies for dealing with it, and that's what many people, including WHO and the Union and other organizations, are trying to work on, to try and build capacity and support countries to cope with this. It's a whole new world that we're having to deal with.
It certainly is, Guy. By the way, do you happen to know whether in Indonesia there are as many markets where you have, the wet markets as they're called, where you have wild animals?
I don't know the answer to that, I'm afraid. Yes, but that seems to be an important initiating factor in this virus. I think at the moment, clearly we've moved beyond that initiating factor to a situation where human-to-human spread is by far the major vehicle for transmission.
Yeah.
But we are going to have to look, when this is all over, we're going to have to, the issue of zoonoses and One Health and the origins of this is going to have to be a major focus for discussion. But I think probably the world at the moment is focused on human-to-human transmission as the major focus for discussion.
Thank you. Next question from Nick in Sydney. We hear a lot about positives and negatives on herd immunity, as it's called. How can Australia best protect our citizens, and will herd immunity be reached with COVID? Gary, would you like to talk about that one?
Sure. So I think we skipped over me before, but I'm currently I'm a professor of genomics and bioinformatics at the I3 Institute at UTS, and our focus is actually One Health specifically, and we have several projects underway looking at the transfer of pathogens from animals to humans specifically. But in respect to herd immunity, the answer is eventually. We have a lot of discussion has been made around particularly the, say, influenza killing more people, but the issue here is that COVID is literally a novel virus, and so human populations currently don't have immunity to it. We haven't been exposed to it in the past. So the actual calculation of how many individuals are required to hit herd immunity for any particular pathogen varies by that pathogen. And you would have heard people talking about the R0 number, which is essentially the number of individuals that an infected individual will in turn infect. And that varies wildly between pathogens. And it seems that COVID is fairly infectious on that scale. Other slightly more infectious viruses would include measles, for example. But this is going to be an ongoing issue for COVID-19, particularly is that we're going to see, I would guess, echo peaks of infection as this gradually moves around the world in and out of populations. And most of the efforts that are currently underway at the moment around social distancing are much more focused on decreasing the impact on the load on the health system. But I would not be surprised to see that the number of individuals required to hit herd immunity will be quite high. But we can throw that out to people with specific epidemiological experience here.
Antonio, did you want to add to that one or the previous question?
I wanted to talk about the previous question,
right,
which was the need for developing rapid testing for COVID-19. And I think, and maybe one of the fail of current technology, one of the way that we have been able to confine SARS, MERS, and similar infectious and actually quite serious diseases was by the use of thermal camera technology. So basically, we had massive screening of people in airports around. And when you see symptoms of fever, obviously you stop and you can help checking whether they have the virus. However, because COVID-19 or the coronavirus is asymptomatic for quite a few days before the symptoms appears, and actually, I think 15 hours before the symptoms appear, it is believed to be the moment where the symptoms are the strongest and you're more infectious to others. We don't have current technology able to tell us how to confine people and how to stop the spread. And so we resorted to basically ancient technology of quarantining everyone home. If we are able to develop technologies to help early stage detection of viral infection, then actually it would be a great step to avoid that things like the coronavirus may happen again in the future. And also to understand how we can limit the spread for the future months, because it's very hard, as we were discussing before, to know what's going to happen when the coronavirus jump from one population, one country to the other in the next month.
Yes, we had a question. Please go
on. May I add to that? Look, the evidence at the moment from around the world is that patients are most infectious on day 4, 5, 6, 7, and 8 after being infected with the virus. However, the finger prick test and some of the other tests that Bill may want to talk about don't start testing positive until a bit later. So we really do need an early rapid test, as Antonio said, so that we can actually pick them up. But the current one will only pick you up at about day 7. So you've now had 3 days of going around potentially spreading it without realizing it because your symptoms are fairly mild. And that's why we need to rely on social distancing. So I would please implore the public that if people aren't in your family bubble, then keep your distance, particularly if you think you've got symptoms, mild symptoms of even a cold or influenza, because you may well have a very mild case of COVID, although the numbers in the community at the moment are from community spread, from unknown community spread is probably very, very, very small. But just be very respectful of others because we can't test you yet. Thank you.
Yes, in fact, the question which we didn't actually read out from Amanda in Auckland went, is there a need to develop a more rapid diagnostic test other than the current PCR, which as I remember is polymerase chain reaction, for which a rather wild scientist got the Nobel Prize, PCR testing that is being used for COVID-19. What testing methods are currently being used in developing countries? Romaric.
Yes, sure. So I think the reason it is so complicated to develop fast and cheap diagnostic tests at the moment is twofold. The first one, we need tests that are very sensitive. We would like to be able to detect a single virus, right? So we don't want to wait until there's a massive viral load in a patient to be able to detect the virus. The second reason the tests are so complicated is they have to be very specific. As Bill said before, there are a number of coronaviruses, but we want to be able to detect COVID-2, not the other coronaviruses, because they obviously cause different symptoms, different types of diseases have different outcomes. So yes, it's absolutely essential we develop fast and cheap diagnostic tests, but these are the two challenges: sensitivity and specificity.
Thank you very much indeed. Next question, virus severity in Australia. Although we have a significant ground number of cases of COVID-19 here, we do not seem to have similar ratio of deaths as the rest of the world. Any reason for that? And the question comes from Alfredo. Who would like to take that one? What about the ratio of deaths?
So maybe a comment from Mary Louise.
Bill.
Oh, sorry, Bill. I was just going to say in Italy, the death rate is enormous because they have an unusually high old population in northern Italy, and they only needed one comorbidity to then push them into severe and critical illness. We don't have that elderly population in Australia. We also don't have an enormous number of our elderly who have one comorbidity. Sure, we have some, but in Italy it would appear as if it was even the mildest comorbidity. Also, they had a very high surge onto their healthcare system, and they were overrun with patients needing ICU treatment. So it became very difficult to control and care for critically ill patients. And at the moment, we don't have that surge. Over to you.
Well, before you do that, Mary Louise, the question I sometimes hear is rather rude remarks about Italian culture. But in fact, their health system is very, very good indeed, is it not?
Their health system is very good, but and their lifestyle is delightful. However, they had an enormous surge, and at one stage they had only I say only 2,000, but they had 2,000 cases, of whom 56% were hospitalized. And of those, about half of those were trying to be in intensive care or critical care. So you can imagine the surge and the enormous difficulty of providing the care to the northern Italy all at once. So then they had to make it a national approach. So we don't have that. So yes, they've got a good healthcare system, but not when they're surged with 1,200 patients all at once.
Thank you. Bill.
So again, to echo Mary Louise's comments, a couple of reviews have been done medically, and particularly looked at German rates, which is of course next door to Italy, of mortality, which are sort of 0.5%, and much higher in Italians. The mean death rate, the mean age of those who died in Italy was 78.5 years, so quite elderly. Secondly, there was a large influx of infections around Lunar New Year because Italy did not close its borders. And thirdly, it has an excellent health system and an excellent diagnostic system, and we're working quite closely with them. And what they've learned from ICU is being shared, particularly with ICUs in Australia. And it does appear that a very large dose, so if you have somebody coughing right in your face, so you get a very large dose of virus and you get it introduced in a manner that gets down into your lungs, that's a very high risk. And of course, one of the early deaths in China was the doctor who described SARS-CoV-2. He was only, I think, 32 years of age. Almost certainly he was getting recurrent high-load virus infection. And it comes back to a lot of the comments. You don't just need one virus sitting on your face, which you touch into your eye. You probably need quite a lot of virus. You need it in a manner that is infective, so not just the RNA, but the whole viral particle. And you need it introduced somewhere that it can cause infection. And that's not just probably on your face, but it's probably getting well into your nasopharynx, and certainly if you introduce it into your lungs. And all of those characteristics were present at some time in many patients who've died in Italy, and many, in fact, healthcare workers who've suffered from infection and subsequently died. That's been often present. So I think you have to have all those factors present.
Jane, may I talk to you? I would like question 17 back. I can't quite see it, so we can go to that. My system's jumped a little bit. There we are, going down. And this is one I'd like to offer to Subhas. What should be our new normal, Subhas, in terms of surveillance and population behaviour? Should we be wearing, in other words, more face masks in Australia? Should we be allowing more personal tracking data?
I think after the government new regulations that not more than 2 people assemble at one place, I think the population behavior will be quite different. But in terms of tracking, of course, we can of course use the smartphone. We can use the smartphone for tracking the person, especially the person infected with COVID-19. So that will be very useful. But in terms of the wearing mask, I think maybe it may give one kind of sense of security. So if that comes in the sense of security, then okay. But otherwise, the mask, if we don't use the surgical mask, then we cannot actually stop the virus going from the infected persons. So I really don't know whether that will be helpful.
Here's one for Antonio on pervasive connectivity technologies. What role do we see for these sort of technologies in the development of detection, testing, diagnostics solutions for CoV-2, for Antonio?
Well, this is actually an excellent question. And I think perhaps we can look at it a bit more from the outside and see that the COVID has exposed one critical technological issues that we are facing in our medical technologies at the moment. And this is how can we do delocalized, fast, low cost, and reliable detection. If you think how much time we spend to find out what kind of disease we have, even when we go to a GP or the medical doctor, often we have still to do quite significant blood analysis. Yes, be sent to a biomedical laboratory, and then we get results back in a couple of days, right? So we have always done this for the past 50 years, maybe more. So where is technology at now? So there is the potential, thanks to the development of nanotechnology, sensing, and also much more widespread utilization of artificial intelligence to start having a diagnostic which is personalized, wearable, and point of care. I think that this is not only possible, but is more and more necessary, because viruses like the coronavirus were somehow bound to happen. If you read a little bit the news, people will tell you that actually a disease like disease X or something was bound to happen, and this is just statistic. We are many people in the world. There is a certain probability of virus to mutate, to be transmitted from animal to the human. And so eventually we would have had a virus, unfortunately, like the corona. So I think this is also an opportunity for us to step up our diagnostic technologies and start personalizing and connecting this together to an actor that would enable us to act rapidly and confine this disease before it's too late or it's getting too late, like the coronavirus.
Next question. Is it seasonal? Some experts claim it is like a seasonal flu spreading well in winter. If so, can we predict the virus spreading faster in coming days in Australia? That's from Shrida in Sydney. Who'd like to take this one? Seasonal infection. Bill.
Okay, so yes, it is a seasonal infection, we think, but of course we haven't yet been through a whole year in any part of the world with SARS-CoV-2. The graphs that I showed you from 2007 suggest that the other coronaviruses are seasonal, although if you go to a place like the Northern Territory, respiratory viruses are present throughout the year, and influenza, for example, causes infection and deaths throughout the year in the northern part of Australia. So it does depend upon your climate as to whether respiratory viruses are seasonal. The second thing is whether this SARS-CoV-2 will persist. There seems to be an assumption going forward that it will, and certainly with the large numbers, you would expect recrudescences in time. However, with SARS previously, of course, that came much smaller numbers, 8,000, and then disappeared completely, with the only infection subsequently being laboratory derived. So I still think it's an open question. If it continues, then it is likely to be seasonal based upon what we know from the other coronaviruses.
But Bill, it seems to be global. It's everywhere.
It is global, and certainly SARS was involving every continent except Antarctica. The big difference seems to be transmissibility. And one thing that happens with viruses is they progress through an outbreak and subsequent pandemics is they tend to become less virulent. They may maintain their transmissibility, but they tend to become less virulent because that's a reversion towards a more sustainable evolutionary pattern. Now, that's what we'd hope. I live in hope. I'm not yet convinced we have enough data to give the definitive answer.
Thank you. Next question, it's one possibly for Subhas. In this era of smart cities, Internet of Things, for example, smart sensors and AI, can we be able to develop and deploy smart sensors in public spaces or masks with sensors embedded, in other words, smart wearables that could detect viruses and can assist in early detection of outbreaks to render efforts towards better management of similar situations in case of future potential outbreaks? Would smart clothing help us, in other words? Savas?
There is possibilities, but practically whether that will be feasible, that's the challenging questions we have. Another important thing is that even though the smart clothes we wear, that need to be very, very sensitive. So the challenge what we have here is that once we collect the samples and we test, unless we have the very special detective system, it won't be able to detect the COVID-19. So whether it's any kind of virus, wearable clothes may not be too sensitive to detect the virus level. It can be any other pollutions, it can be any other bacterial infections, but it will be very difficult to have wearable clothes to have that sensitivity to be used by general public in the smart city environment.
Ron, would you like
sample testing will be the better compared to people wearing the clothes for detecting that level in the smart city environment.
Smart answers. Ron, do you feel like answering that one as well?
No, I don't have anything to add to that.
Right, okay.
Maybe I can add a couple of things.
Yep.
So I think this is a central challenge. Can we develop maybe not wearable clothes, but wearable technology, electronics that can help us track in a not real time, but in a sufficiently frequent way viral infections, and which viral infection should we go after? And I think the convergence of nanotechnology, chemistry, and physics, quantum physics may help actually reaching sufficient selectivity and sensitivity. The question is, however, what should be the cost? Can we do it for the West population? And how then to mass produce this? But this is actually a key challenge many people around the world are working on at the moment.
Yes, but how would you react to that? Imagine you've got some smart clothing on and it goes ping, you've got a virus. How is the ordinary citizen supposed to respond to that apart from panic?
Well, actually, this is an excellent question, Robyn. In addition to my research work, I also am head of a strategic research initiative of the Australian National Technology. It's called Our Health in Our Hands. And basically, it's both producing new technology to measure virus infection and other diseases, but also how then use this. Because how would you use this overwhelming amount of data that what would you do? Like, okay, I have a virus. So actually building the interface, artificial intelligence, and connection to our health system, GP, is also a critical step to use this properly in the future.
Nathan, do you want to add to that?
No, I think that's a very sensible response. Nothing to add.
Fine, thank you. Let's go to epidemiology. If no vaccine was found, could we ever see COVID-19 completely disappear through the isolation and social distancing techniques? That's
I guess I can say something to that.
Yeah.
I mean, you need to understand that social distancing is not designed to eradicate the virus. It's merely a mechanism to slow the rate of transmission and to flatten that famous curve we always hear about. The level of social isolation required to completely eradicate the virus and the time needed for that to occur would be completely impractical, not feasible for society to sustain. So it's not about eradicating the virus. It's simply about reducing the rate of transmission and flattening the curve.
Gary, do you want to add to that?
Can I just make a comment about immunity, which has been quite topical with the term herd immunity? If these coronaviruses would result in a sterilising immunity, then that may well, with sufficient infections, result in a reduction. Although if you look at vaccine studies where you try and have a ring of vaccinated people around susceptible individuals, for example, you need about 80 or 85% or more vaccination. And so you would think in terms of herd immunity, you'd need very high rates of infection in order to do that with a transmissible virus like this.
Next question about shoes. And may have missed it. I don't think we have talked about shoes very much. What is the position regarding the cleaning of shoes? Because the virus seems to hang around for a long time on materials, with no recommendations of shoes so far on other social media. That's for Mary Louise, perhaps?
Hi. Okay, so as I mentioned a little while ago, there was a very small study looking at healthcare workers looking after COVID-19 patients, and COVID-19 virus was found on their shoes. But can you imagine that these patients are shedding a lot of virus? So they now will be wearing shoe covers that they'll be replacing so that their shoes aren't contaminated. But for the question in the public, it's highly unlikely, unless somebody coughs on your shoes, that you'll get the virus on your shoes, because of course it has to come from your lungs or your nose. So they will have to be coughing onto you. So you will know if your shoes are potentially contaminated. And if you're concerned, wipe them over with soap and water after you've gotten rid of the dirt, the visual dirt. And you're probably less likely to want to use a bleach. Of course, 65% plus ethanol works as well, but just soap and water if you're very concerned. That's all you need. Over.
Thank you. Still on hygiene and antibacterial wet wipes. Do they, the ones we buy at the supermarket and claim to kill 99.9% of germs, do they work on COVID-19? Pali, what do you think?
Well, it's not a simple answer, unfortunately. Yes, in principle, they should work because they're usually loaded with ethanol. In practice, it's again about all the hygiene techniques, how you use them. will you really cover the entire surface of your hand? Do you miss any bits? That's the key thing. And wipes, personally, I think are probably the least preferred option. Gel, antibacterial gel would be better, and soap and water is best, simply because for the untrained person, it's easier to do that movement and getting everywhere. Now, I did also find recently an old study on influenza viruses where they did compare different wipes and just on surfaces, and they said that some antibacterial wipes killed the influenza virus. Oddly enough, not all of them. So this hasn't been done on COVID, obviously. So yes, use the wipes if you have nothing else, but I wouldn't use them as the frontline protection. I would use soap and water or alcohol-based gels.
Anyone else? So let's move to Louise.
I was just going to say, most of the ads that say they kill 99.9% of germs are correct. They kill the germs, but they may not be pathogens. And certainly no wipe has been tested and approved by the TGA that will kill COVID-19. But Paula is absolutely correct. Most of these wipes are full of an alcohol, and that should work as long as it's at least 65 to 70%. But soap and water, as Paula said, is superior. Over.
Now a question perhaps for Bill on comparative epidemiology of diagnosis. There's lots of punditry based on league tables comparing rates of both infection and death in different countries. What are the main pitfalls of comparing Australian death rates from COVID-19 with other countries?
So some of the pitfalls I can think of, and I'm sure there'll be others who can add, are the knowledge about background rates. So how many people are we testing? So what proportion of the population? And how many of those are positive? That gives us an expected rate, but of course there's all kinds of biases in that. That may be a population that's more wealthy because they can present, maybe a population that is less ill, that is, or they may be more ill because there may be a large population, particularly of children, we believe, who have mild or effectively asymptomatic illness and are therefore not being tested, and therefore we don't know our denominator, we don't know that baseline. Thirdly, it's going to depend, of course, upon your test. In Australia, we have very good diagnostics, but this has all been introduced very quickly, and so we don't necessarily have the kind of denominator information that we might get from having parallel tests with things like old-fashioned virus culture, or, as has been mentioned, antibody tests, or indeed even more sensitive detections with things like nanoparticle or smart sensors. Thirdly, the actual virus may vary. There's been a lot of discussion whether there's a different virus, for example, in Italy causing more deaths than in Germany or Australia. There's been no evidence for that so far. So there's the overlay of wealth, if you like. Which populations are presenting? Are they, in their terms of their demographics, you know, they're wealthier, are they younger, are they more men, more women, all those kind of things. So all that determines your denominator, and therefore it determines, you know, the ideas about rates and your understanding of that.
Yes, and that question was from Declan, University of New South Wales. Thank you.
Can I add one small
question, Louise?
Robyn? One of the problems with developing a death rate during an outbreak is that when an outbreak is highly volatile, usually outbreak epidemiologists don't try to establish a death rate, because what you have to do is wait until the cohort has had time to recover or die. Now, all of the problems that Bill has said is absolutely right. So you've got a potential biased population between the countries, but you also may have a large number of people that are going to take quite some time to get well or die. So what you're doing is developing a rate that is not correct. So if you actually do an adjusted one to begin with, it's not perfect, but you divide the number of deaths by the number of recovered plus deaths. That'll give you a vague idea of a small cohort in time to work out what the death rate is. Another way of doing it, and it's still not perfect until waiting to right at the end of the outbreak, is to count back about 2 weeks if you think it will take somebody 2 or 3 weeks to die. So what the average period of time from diagnosis or developing onset of illness to death is, count back and identify what the total number of cases were then. And that's their cohort, and use that to develop a death rate. It'll still be higher than 1% or 3%. So, for example, in Wuhan, the death rate is much higher than the 1% or 3%, but eventually we'll know what the true death rate is.
Interesting.
Can I just add something, Robyn? I think I think that the other thing that was suggested by Bill was it's going to be important fairly early on to start doing serological surveys of representative samples of the population. So serology is going to give us the best measure of the true underlying incidence or cumulative incidence of infection, and that will be a better basis for comparison. It'll also be a good basis for telling us how much undiagnosed infection is going on in the community, and a good basis ultimately, as Mary Louise says, you need to wait time, but ultimately for looking at what the case fatality rate is.
And Gary, how does that work? Do you take blood samples or what?
You do blood samples, yeah, but it needs to be of a representative sample of the population, and it's offset by about 2 weeks because it takes about 2 weeks for the serology to become positive. So you're really learning about what happened 2 weeks before you did the serological survey. But
and
that's
you get a true picture of how many people have got the bug.
Have been infected, and you also get the secondary piece of information of how many people have antibodies that are likely to protect them from being reinfected. So you get some picture of the progress of herd immunity as time goes on, and you could do that on a serial. You can do that on a serial basis and track the progress of the epidemic. It'll always be difficult to track the progress of the epidemic from notification of virus isolation for all the reasons that have been discussed, because we don't know about the cases that we're not testing.
And a question, I'm reminded from what you said about some work by McClure and Burnett. How does the body learn how to make antibodies for a germ it's never seen before? I mean, it's bloody clever, but how does it happen?
I'm happy to field that question. How does it happen? It's called adaptive immunity. So the issue we have at the moment is there is no memory immunity, and that derives from adaptive immunity, and that's mediated primarily by antibodies, your protective memory immunity. And so that takes time to develop, and that a couple of weeks for that to develop, and that's why it takes some time to get the serology. And in those couple of weeks, you're essentially completely vulnerable to the effects of the virus, and you rely on another part of your immune system called the innate immune system, which essentially tries to contain and control the infection, keep you alive whilst you start getting your adaptive immune response up and running. And as I said, that takes time because that involves recruitment of multiple different types of immune cells, presenting bits of the virus to the immune system, selecting certain cells that are going to respond to those bits of the virus, expanding those, then getting them to generate sufficient amounts of antibody to be therapeutically or to start eradicating the virus and so you can recover. So in a nutshell, it relies initially on innate immunity, activation of adaptive immunity, and that just takes some time.
Well, I think it's one thing that Australia can really boast about, because what you just said is the most astonishing achievement in science. And McClure and Burnett was actually called a genius of the 20th century. You know, Sir Mac from Melbourne, working that out with Peter Medawar.
Yes.
Who actually came from Argentina but lived in Britain, and trying to find out how an innocent body can then learn, decode, and get a response that makes you able to resist this new disease, I think is one of the most phenomenal achievements, both intellectually and of your bloody body.
Yes, and Peter Doherty also needs to take a considerable amount of that accolade as well for understanding the role that sort of T cells play in that process, because it's both B and T cells working together to give you the antibodies you need to ultimately eradicate the virus and enable you to recover.
Yeah, T cells being the thymus cells.
T cells mature in the thymus and then leave the thymus once they've been selected to attack a specific pathogen.
Did you touch your thymus while we have you on camera?
The thymus is just above the heart.
That's right. It was the last organ to find function in science, and that was discovered in Melbourne.
Indeed.
Extraordinary. All right, end of a bit of history, but let's move to a question from Nate. If there is evidence that food allergies can cause inflammation problems, would it make sense that diet could have an impact on making the systems worse? Rom, perhaps?
Sorry, I think this question is not for me.
It's not for you.
I think that one is directed to me as well.
Yeah.
And look, it's a very important question, understanding, I guess, comorbidities, other diseases you might have when and if you get COVID-19, and do the other diseases have the potential to make COVID-19 worse for you? In terms of allergies, I don't think there's any evidence to suggest that certainly a food allergy is going to make your COVID-19 any worse. Even with allergies of the airways, such as asthma, it doesn't appear that these also increase the severity of the disease. So I think in terms of interaction with other diseases you might have, there's no evidence to suggest that certainly allergies are going to make it worse. The other thing to consider is treatments for those allergies. You may be on immunosuppressive medications for allergies or autoimmune disorders, and there may be a possibility that those medications immunosuppress you and also potentially make the infection worse. Again, we don't have enough data on this to understand how that might happen, but it's certainly something we've got our eye on.
So it's Guy here again. So the early data at least suggests, as Nathan says, that people with asthma and with underlying lung disease are not, in fact, at particularly increased risk from this disease, which is a little surprising, but that's what the data seems to be showing. The only diseases that are clearly related to increased risk have been diabetes and hypertension. And there remains some uncertainty as to whether that increased risk in people with diabetes and hypertension is a consequence of the diseases themselves or of the treatments for those diseases. There are treatments that many people with those diseases take that could alter the response to the virus. This remains unknown, basically. The recommendation at the moment is that everybody should continue to take their regular treatment for diabetes and for hypertension, but it's an area of active investigation as to why it is that people with diabetes, high blood pressure, and possibly other heart disease are at increased risk of adverse clinical outcomes from this disease.
Thank you, Gary. Anybody want to add? In that case, let's go to question 27. We've done lots, haven't we? Graham in New South Wales. What is the minimal level of filtered mask? In other words, what number that will protect you from COVID-19? Does it need to be equivalent to a surgical mask level? Paul, do you have an opinion on that?
Yeah, so there are two parts to this. I mean, there is, when we talk about sort of face protection, there's the mask that you wear to block out particulates, but also in conjunction, it's recommended you wear a face shield as well. In terms of the mask, actually the requirements are that if you're in a high-risk area, you want to have better filtration than a surgical mask. And in Australia, that's the P2 mask that's designed to keep out particulates. It tends to be a combination of different layers in the mask designed to both physically block particles, but also some of the layers remain electrically charged. They actually have an electrostatic charge that helps attract and filter out the very small particles such as viruses. The face mask itself then, or the face shield rather, goes over the top of that and is designed to block the initial spray or spatter of someone's coughing in the faces of, for example, a medical professional who's treating that person. I don't know if Mary Louise wants to add any more in terms of WHO requirements around this subject.
Mary Louise?
Hello, can you hear me okay? Great. Yeah, look, WHO expect all healthcare workers to wear a level 2, what we call in Australia a level 2 mask, which will filter out at least 98% of particles. So there are ones that filter out 95%, and look, they would be perfect in the community. The likelihood of you coming across somebody that's going to cough at you is minimal. So in Australia, where healthcare workers are looking after people shedding, it needs to be at least and over 98%. So yeah, that's for the surgical mask, and I think you've answered it very fully. Thank you.
Thank you. We've got lots of questions waiting, so let's move on quickly. 28, elimination of the virus. Can the virus risk ever be completely eliminated from the human population if transmission was completely to stop at a point in time? From Ian. Guy, do you think we can get rid of it completely?
I think others have addressed this before. I think it's unlikely we will completely get rid of it. It has been possible with some viruses to stop transmission, and you know, with the initial SARS virus, with Ebola, in fact, you know, transmission of the virus has stopped, but the risk that it will come back is very great. And I think the extent of spread of this virus already means that it's very unlikely that we will completely eliminate this virus without a vaccine. I don't know whether others want to make a comment about that.
I can make one additional comment to that. I think this is why diagnostic testing is not only important in humans, but it's also very important in animals, other liquids in the environment. We have to find all the reservoirs, all the places that the viruses are hiding in before they could be picked up again.
And the other issue, yeah, and the other issue will be how effective the vaccine. If we look about the viruses we've eradicated through vaccination, talking smallpox and polio, essentially are probably the most famous ones. And that's because the vaccine is really effective and the virus doesn't tend to escape the vaccine. With respiratory viruses, there's huge diversity within even related groups of viruses. So what the real challenge is going to be is how good the vaccine will be and how able the virus is going to be able to escape the vaccine, and for versions of the virus to continue to circulate that the vaccine doesn't protect.
Next question from Ayu in Sydney about recovery and reinfection. What do recovery from the virus look like? What does the recovery look like? And will it leave permanent damage? She says, I've heard Ayu anecdotal accounts that it leaves permanent damage to your lungs and reports even of reinfections. Guy, what do you think?
Well, I think it's a bit early to know this for sure, because this will require some study of people who've recovered from infection, which really we haven't had an opportunity to do. But what we know from other respiratory viruses like this is that most people do, in fact, recover completely from the virus, and it would not be expected to leave long-term consequences, except in the small proportion of patients who get this very severe problem, acute adult respiratory distress syndrome, or ARDS, that the X-ray picture that Bill showed right at the very beginning, people with very severe lung disease, those people may well have long-term sequelae, long-term consequences for their lung function beyond the disease. But that's going to be a very small minority. I think that the majority of people will make a full recovery. The issue about susceptibility and the likelihood of being reinfected is really a completely separate issue, unrelated to damage to the lung, and that relates to the issue of how effective is immunity. And I think Bill and Nathan have already discussed that, and we're not clear yet how effective naturally acquired immunity will be.
Anybody want to add?
I'll just add the issue about reinfection is one that I think is an open question. Most of the cases that have been written up that I've seen have actually been detection of RNA, and we get back to the discussion at the very start. RNA is just the genome of the virus. It's not infectious virus, and so reinfection seems very unlikely, except in a small group of individuals, possibly with immune compromise. So the evidence so far is we're not seeing reinfection. What happens in 12 or 18 months' time is an open question.
Yes. Now we have the chloroquine question at last from Martin. It's obviously an informed question about anti-malarials. He says, I understand that chloroquine has a mechanism of action relating to blocking hemozoin production in acidic conditions to block the replication of Plasmodium for malaria. I can't imagine that the hemoglobin modification of chloroquine would do much for the virus. What would the mechanism behind chloroquine being used to treat COVID-19 be? Would the acidification prevent the envelope folding away from the nuclear capsid for the coronavirus? Who will take this one? There's a very technical,
I'm happy to field this one. This is all still rather speculative, but what is thought to happen is chloroquine it's a basic molecule, and what it's thought to do is inhibit the capacity of endosomes. And just to explain what they are, these are structures within a cell that the virus is initially taken up into as it enters the cell. And often what happens with viruses, they require the pH in the endosomes to drop to become acidic, and this changes the conformation of the structure of the virus and allows the virus genetic material to be released and to initiate the process of replication. And what is thought to happen with chloroquine is it's supposed to inhibit this process by preventing that acidification of the endosome, stopping that conformational change in the virus's structure and allowing that initiation of replication. So that's what's thought to happen. This was still very unclear exactly if that's the case.
May I just ask you a rather tricky, provocative question? What do you think of the multi-age adverts this week from a certain Clive, a billionaire,
We just come up, didn't we?
suggesting we best produce this stuff and hand it out?
Yeah, look, I guess others can probably speak to this as more than I can, but I think we sort of had a quick chat about this before we went on air, just because we knew this would be an issue that would come up. Look, there is, as I said, some evidence that it might work clinically. I guess the key thing here is it needs to be utilized very carefully, and we wouldn't be condoning it just to be handed out like candy for people just to self-administer, because it does have side effects and it can be toxic and indeed lethal if used in high doses.
As Louise was saying earlier.
Yeah, indeed. So, I mean, I'm happy to hand that over, please. I don't know.
Perhaps I might just add that there are already a number of trials ongoing of this drug, and I think it's very important that we allow those trials to run and that we support them, and that we try and set up mechanisms within Australia so that everybody who has the virus actually participates in clinical trials to test the effectiveness of this and of other putative treatments, so that we learn as quickly as possible what works and what does not work, and patients can get evidence-based treatment for the virus, or evidence-based treatment for the virus if it turns out that there are no effective treatments at the present time. Sporadic use of viruses on sort of of drugs on anecdotal evidence is the worst thing we could be doing at this time. We need evidence.
Do its work. Next question from Richard in Sydney. He says, recovery time needed. What is the time required for an infected patient to recover fully on average? And how do we shorten it significantly? Sascha. Yes.
Oh, great. So I think this is a really good question, and probably difficult to answer because it would be variable. It depends on a person's immune system and their body's ability to respond to the virus. But we know that from infection to developing symptoms is about 3 to 5 days. And again, that is variable as well. But I would think that most healthy people would recover by that sort of 2-week period of the 14 days that we've seen. But I'm happy to flick that over to Guy or Bill or a clinician for that sort of advice. And how do we shorten it significantly? I think we've just talked about various drugs that we can use to perhaps antiviral drugs that we can use to target the virus, which we sort of don't have at the moment, but everyone around the world is madly working towards. So,
anybody want to add? Okay, let's move on. From Susan.
May I add one small thing, Robyn? Is that from overseas experience in particularly China, we found that if people wait too long to get to hospital and Italy, if they wait for about day 12 and they're hospitalized, they have a more severe case and they don't do very well. So I would really require the public to start thinking about how well they're going, keep an eye on their health and go to their GP, particularly if their symptoms continue, because I know that they're often not being able to get a screening test or a diagnostic test. Because don't wait too long if you're 60 and over, and if you have a comorbidity, certainly don't wait 2 weeks before you get seen by a doctor. Over.
Thank you. Now a question possibly for Gary from Susan, who says merging data sets in Taiwan. Taiwan has handled the pandemic more successfully than most countries by tracking cases. For example, the databases from immigration and health were merged. How can Australia improve its tracking capabilities before it's too late?
Well, the big issue in Australia is that we're not doing enough testing. And there's a variety of reasons for that. A lot of the countries that have been very successful at controlling this have done a great deal of testing on a large number of individuals that has enabled contact tracing and then a lot more aggressive measures once they get positives. At the moment, Australian testing is fairly restricted to those who are doing, who are coming back from overseas, for example, and so we're not really capturing the community spread. So there are tests starting to come out. I see that Abbott Laboratories have come up with a rapid diagnostic test, which is what the clinicians desperately need to be able to have a yes/no test, so something basically a highly specific test. We probably don't want to worry too much about false positives. We do want to worry about false negatives. From an epidemiological perspective, what we want is a lot more whole genome sequencing of these viruses to be able to completely or more accurately map how the virus is mutating, if at all, and who's transmitting it. So the virus is running at around just a touch under 30,000 bases in size, which is tiny when you consider the human genome sitting at around 3 billion. But that's still difficult to do quickly and in large numbers. And that's actually one of the things we're trying to do at the moment, is to do high throughput sequencing of full length virus, thousands to potentially millions at once. But that's essentially what we're going to need to have going forward, is rapid tests that are highly specific that can be rolled out to population level testing. And Australia, of course, has the challenges that, say, the United States have of a large dispersed population who may not be as, say, amenable to some of these measures that, say Singapore or Taiwan are able to be done.
Yes, we have now just time for 2 more questions. This one from Don in Canberra. Would it be better to set up separate COVID-19 treatment facilities away from our hospitals, better for staff, patients, contagion, infection control, etc.? Use a disused building, tents on an oval, etc. Neil, what do you think?
It's a good question, and certainly in places like China and Italy, that's been done as needed. The reasons we wouldn't do it at the moment are our high-level therapeutics like ICUs, like expertise are concentrated in hospitals. Whether that's right or not, it's a reality. And secondly, the testing is based very much around doctors and referral through GPs, as well as through fever clinics, which have been mentioned. And thirdly, we simply don't seem to have the need at the present time. As I said before, I live in hope. Hopefully we wouldn't need that. In terms of infection control, we have very, very highly adapted and developed infection control systems within hospitals, and that includes places like ICU and the fever clinics. If you have a large dispersed population, certainly you get distancing, but then you have to have a large number of people to service those people medically. And of course, those people have to have proper infection control training, and it's not all that simple. So I think it has been done where it's been needed. Unless it was needed, it wouldn't be something that would be a preference from my point of view as an infectious disease clinician.
Thank you. And our last question, which is one for everybody, something everyone's interested in, from Marianne. How long will all this last? Is it too early to predict when the pandemic will leave Australia and some sense of normality will come back? Mary Louise first.
Oh, so this is probably the most difficult question to answer because there are so many things we don't know about this virus. But I've been looking at the data, and the doubling time has slowed down. At one stage, it was doubling every 3 days. That has slowed considerably, but the numbers, the new cases haven't. WHO call an outbreak at an end, that it ceases, when we have a last case and twice the incubation period. We are nowhere near the last case, and so we are nowhere near the end. So please don't stop social distancing and hand hygiene. I think it'll be a long time yet. The other reason it'll be a long time yet is because the data are at least 7 days old. We're like as if we're in the Doctor Who TARDIS and we've gone back in time, because the data today represents what's been happening about 7 days ago with incubation. Then we have the delay of the testing results, but that will change with new testing methods, and we'll get much rapid understanding of what's happening right now today when we open up our Excel spreadsheet. So I'm sorry to say we're nowhere near the end yet, but we are at least slowing down. But hopefully tomorrow that will continue to slow down to double every 5 or 6 days, and then we may be on a trend, because it's not just a day or 2 days. We look for a trend, and sometimes you can get spikes when accidentally you get the Ruby Princess coming in, and that spikes the numbers. So
that's all we have time for, our final question, and thank you so much for sending so many. It's been really wonderful, that range. I'd like to wrap up by giving an idea of history of where we might be at the moment. And it takes me back nearly a couple of 100 years to a place called London. And John Snow, a doctor who helped save one of the most important situations imaginable, which was a plague, cholera mainly, and he examined what was going on, why some people got sick and other people did not. And he famously took the pump, the handle off the Broad Street pump, and as a result of that, the germs failed to flow to a certain area. And that act of genius helped invent modern epidemiology, the study of the way diseases travel. And the next person who took up the amazing role was in fact an architect in some ways, Joseph Bazalgette, who invented the sewers of London London was an absolute cesspit. It was so foul that they had to evacuate Parliament House by the Thames because it was so stinking and vile. And as a result of what he managed to get through doubting people all around London was this amazing sewer system, which in fact not only cleaned up London, but actually gave the world a model of what to do to save lives and in fact make a better place to live. The whole embankment was invented by what Bazalgette did. So here is a situation now in 2020 that might be the beginning of a transformation of society right around the world because of this incredible, nasty disease. Thank you. Justin, over to you.
Thank you, Robyn, and thank you everybody for sending in your questions. You haven't heard from me because there's been so many questions. We've been trying to field them and get them into a good order. This will be actually repeated on Sky News in the weekend, and it will also be on the NSSN website, which is www.nssn.org.au. I think I'd like to thank Robyn for donating his time, and you can see why a professional is needed. It's made an incredible success. I'd like to thank the panel and expert audience for sharing their time and expertise. I've certainly learnt a lot. I'm going to go back and look at it again to learn more. I'd like to thank the whole NSSN team who've put an incredible amount of work to get this together in a very quick time. They all know what they have done. And most importantly, I'd like to thank all you out there. We've had questions from Australia and the US. I'd like to thank you for spending your time listening to this forum and sharing your questions. I hope it was useful. Stay healthy, stay safe, and stay positive. Thank you.